At the foundation of sexual reproduction is a cellular process of remarkable precision. It's called meiosis. And for it to work properly, our chromosomes have to solve a fundamental organizational puzzle, one that has been a subject of scientific inquiry for a long time.
You see, inside the crowded space of a cell's nucleus, every chromosome has to find its one specific counterpart, its homologous partner. It's a situation that's a bit like being in a massive, busy room and having to find the one other person who has a key that perfectly matches your own. So this brings up a central question. How is this pairing achieved so accurately, time after time?
And the consequences of failure are significant. If a chromosome pairs with the wrong partner or doesn't pair at all, the resulting cells can end up with an incorrect number of chromosomes. This condition, called aneuploidy, is often the cause of serious genetic disorders. So, this matching process needs to be exceptionally reliable.
To get to the bottom of this mystery, researchers have often turned to a small nematode worm called Caenorhabditis elegans, or C. elegans. It's a well-established model organism for studying meiosis. Their work is focused on a particular protein that they suspected plays a central role in this whole matchmaking process.
The protein is called HIM8. What was already understood is that HIM8 acts as a kind of molecular anchor. It attaches itself to a very specific spot on the X chromosome, a region known as the pairing center. But exactly how this simple binding could help two separate X chromosomes find each other in the nucleus was still unclear.
So to figure out its function, the scientists took a classic approach. They decided to dissect the protein piece by piece. By creating different versions of HIM8 with certain parts removed, they could start to map out what each section of the protein was responsible for.
You see, the HIM8 protein isn't a simple uniform structure. It's made of three main parts. There's a structured beginning called the N-terminus and a structured end, the C-terminus. And sandwiched between them is a long, flexible, and seemingly unstructured middle section, what's known as an intrinsically disordered region, or MIDR.
The critical insights came when the researchers began removing these sections. If they took out either the N-terminus or the C-terminus, those structured ends, HIM8 couldn't bind to the chromosome at all. But when they removed the flexible middle region, they saw something different. The protein could still bind to the chromosome just fine, but the chromosomes themselves completely failed to pair up.
This result was the central clue. It told them that the structured ends of the protein are the anchor, but it's that disordered middle region that is somehow doing the work of pairing. The investigation then shifted to a new question. What exactly is it about this unstructured region that's so essential for bringing chromosomes together?
The answer, it turns out, came from a completely different area of physics, revealing a property that challenged the existing mechanical models of how chromosomes might interact. It provided a whole new way of thinking about the problem.
When the researchers isolated that middle MIDR region in a test tube, all by itself, they observed something quite specific. The protein molecules began to self-assemble. They separated themselves from the surrounding solution and spontaneously formed distinct microscopic droplets. This process is known as liquid-liquid phase separation. You've likely seen a version of this principle in action with oil and vinegar and salad dressing. The oil and vinegar don't change chemically. They just have a stronger attraction to their own kind than to the surrounding liquid, so they condense into their own separate liquid compartments. This discovery represented a major conceptual shift. It suggested that HIM8 wasn't just a static hook holding onto the chromosome. Instead, it was actively creating a physical microenvironment around the pairing center, a dynamic phase-separated compartment. So with this new idea of a liquid droplet on the chromosome, the next piece of the puzzle was figuring out how this structure actually helps with pairing. The evidence suggests that this droplet provides a vital connection between the chromosome and the boundary of the nucleus, which is called the nuclear envelope. This HIM8 droplet essentially acts as a localized hub that attracts and recruits other proteins. In particular, it gathers proteins like SUN-1, which is located in the nuclear envelope. By concentrating these proteins inside its droplet, HIM8 effectively builds a flexible tether that connects the chromosome to the inner surface of the nucleus. Observations under the microscope support this. As a quote from the research paper notes, within the cell, HIM8 and SUN-1 are seen forming these dense clusters. This isn't just a simple one-to-one binding, but a collective aggregation, a behavior that's consistent with a phase-separated compartment concentrating its components.
To test this idea further, the researchers used a chemical called 1,6-hexadiol, which is known to disrupt the weak interactions that hold these liquid droplets together. Sure enough, the chemical dissolved the SUN-1 clusters and broke the tether to the chromosome. Then, when the chemical was removed, the clusters and the tethers reformed. This confirms the connection is dynamic, based on weak, reversible physical forces.
So all this evidence, a protein that binds to chromosomes, a middle region that phase separates, and a dynamic tether to the nuclear envelope, allows us to construct a new, comprehensive model for how homologous chromosomes pair up.
Here's how the model works, step by step. First, the hemate protein binds to the pairing center on each of the homologous chromosomes. Second, its middle region undergoes phase separation, forming that liquid-like droplet. Third, this droplet tethers the chromosome to the nuclear envelope by recruiting proteins like SUN-1. Fourth, the cell's internal motors then move these tethered chromosomes, pulling them along the inner surface of the nucleus. And finally, fifth, when two of these droplets on homologous chromosomes happen to meet, their liquid nature allows them to merge, or fuse, into a single, larger droplet.
This fusion event is the key recognition step. By merging their liquid compartments, the two homologous chromosomes are brought together into a stable, close association. This then allows them to align perfectly and become physically linked by a protein structure and a process called synapsis. At that point, the pairing is complete.
So this research reveals something more than just how a single protein works. It demonstrates a powerful physical principle, phase separation, organizing a critical biological process. And this brings up a much broader question. If these dynamic liquid compartments can orchestrate something as precise as chromosome pairing, what other fundamental processes inside the cell might be organized not by rigid machine-like parts, but by these transient, self-assembling liquid domains?